I want to emphasize with the structure, the DNA structure here, that are going to come back into play a little bit later when we talk about transcriptional regulation. So, right, first level of compaction is at the nucleosomal level. And this adds about a 5-fold compaction level, which is actually pretty surprising, considering that's not really on that big a deal, right? So you're just wrapping your DNA around what we call nucleosomal cores to create the chromatin. So chromatin, right? You've heard that word, I'm sure, a lot in your studies. So the chromatin that's in our nuclei is just the DNA plus the nucleosomal proteins that they're wrapped around. And so the cores here, these little yellow beads, if you will, are composed of 4 different proteins. So it's an 8-mer, it's an octamer, to each of 4 different histone proteins. Histone H2A and B, H3 and H4. So there are 4 different histones, 2 copies of each of them to form the core, which is highly positively charged, right? So think about it. Our DNA is negatively charged, so the nucleosomal core is actually positively charged to be able to wrap this DNA around it. 2 superhelical turns around each core. And the thing that's actually pretty important to remember is that these histone proteins are actually super malleable in terms of how they can be modified. Okay, so they can be phosphorylated or acetylated or methylated. And that is going to very seriously change the way that they're interacting with the DNA and how the DNA is going to be either open and exposed or cranked down on and not transcriptionally active. And so acetylation is something that we're going to talk about an enormous amount for these histone proteins. So let's talk about that. So here we have our chromatin fibers, or what we call a solenoid. Okay, so the solenoid, the base of the solenoid, and this is what I was talking about, the 3D, the electron micrograph structure of these guys. Really quite stunning. And the chromatin core, this solenoid, has 6 of these nucleosomal subunits in a turn. Okay, so remember, we're always turning. DNA, I don't know how it doesn't get dizzy, right? It's turning. A helical turn of just the regular old DNA, and then you got superhelical turns around the nucleosomes, and then you got turns of the nucleosomes on each other. Okay, so spinning. Lots and lots of turning going on here. So each of these nucleosomes, right, has histones in it. They are positively charged, right? Each of our histones are positively charged. And the nucleosomes in general are positively charged. And so what can happen is that how do you get these solenoid coils, right? How do you get all that DNA wrapped up on each other when it's all negatively charged, right? Well, what happens is you've got crosstalk between the DNA, say, on this nucleosome here, and this nucleosomal protein over here, these histones over here. So you've got kind of interlacing and crosstalk between the nucleosomal structures so that the histones in one nucleosomal core can interact with the DNA on another nucleosomal core and keep it all compacted together. You also have the addition of another protein called histone H1. So H1 is not part of the nucleosomal core, but it's involved in this compaction process because it's actually bound in the areas that are coming in and out of the nucleosomes. So if you look at this for a second, right? So H1 is bound up on these intermediate structures. Okay? So H1, again, is a positively charged protein, and so it is also helping this compaction of the nucleosomal cores together, because H1 over here can help to bind to the DNA, again, on another nucleosomal core and help to compact the structure. Now, we said that these histones can get modified, right? So acetylation is one of the key modifications that we're going to look at. Acetylation onto the histone proteins, particularly H3 and H4, is going to impact the charge of that nucleosomal protein so that it actually becomes negatively charged. Okay, so acetylation negatively charges that histone molecule that is on. And right now you're adding a negatively charged molecule into this structure that's really super compacted. Now all of a sudden, you're going to start getting repelling charges. Okay, so if you acetylate the nucleosomal core on this nucleosome, well then all of a sudden you're going to start repelling the DNA that's over here on this nucleosomal structure. So you're going to open up the structure. Okay, so we're going to relax this core structure. We absolutely have to do this in order to get the replication and transcription machinery into this molecule, into this solid. Okay? Yeah.
I think I just got a little bit lost in that. So
Okay.
when you acetylate it, are you acetylating the histone, or what is it that you're...
Here, acetylating a histone.
Which gives it a negative
H3 and
charge.
H4, which gives it a negative charge, which is now going to repel it away from this solenoid structure and the DNA that's on the other nucleosome.
So
It's also going to kind of repel the DNA that's on it as well, right? And that's what's helping to loosen up the structure as a whole, right? Because you've got to get rid of that nucleosome in order to actually replicate or transcribe. You got to get the nucleosome out of there as well. So that's helping to relax the structure. Very good point. Does that make sense, everyone? So you're not just repelling the DNA that's on the other nucleosomes, you're actually repelling the DNA that's on you yourself, so that it loosens up the structure so that the transcriptional machinery can get in there. So this is, hold on one second. So this acetylation process is hugely important for transcriptional activation. So we're going to see this acetylation happening all the time when we talk about transcriptional activation, when we talk about epigenetic regulation. So keep that in mind. This is an important modification that we have. Josh?
Can the acetylation be heritable? Maybe passed on?
Not acetylation, methylation. Yeah.
These are just occurring on the side chains of amino acids.
What is occurring on the histone residues? I can't off the top of my head remember which residue it occurs on. But it's on a chain within the histone protein itself. I can't remember what residue it is that it modified. I think it's lysines, but not 100% sure of that. Okay, so it makes sense. So compaction and then release, right? So think about that. The DNA inside of our nucleus is really breathing, right? It's a malleable structure. It's not just here it is, bing, bang, boom. We're always going to keep it in exactly the same conformation. This thing is moving a lot and being modified a lot when we need to replicate, when we need to transcribe. Okay. Right. So lastly, I want to talk just a little bit about the repetitive sequence that we've got in our genomes, because it's insane. Oh, and I also want to point out that this is a different slide than what you have, because I realized that I loaded up the year before's PowerPoint, so I'll put this in so that you have it. Sorry about that. I notice little things as I'm going through these at night before I come in with you. And so you might that might happen. No worries, you'll get the latest, greatest versions of these. So, right? We talked about this. Our haploid genome, right? And when I say haploid genome, I'm talking about the content of a single set of chromosome pairs, right? So if we have 2 copies of chromosome 1 and 2 copies of chromosome 2, if you have 1 copy of the DNA, that's your haploid genome, okay? So our diploid genome, all of the DNA would be 6.4 billion bases. Okay, and as we said, 3% of that is actually single copy genes, protein coding genes. Coding genes. That encodes about 21,000 gene structures. And here's the fun fact, right? We are, to each other, 99.9% identical, right? So why do we all look so crazy different? Well, we don't really all that much. Little minor details, but you know, we've basically got the whole encoding structure. And here's the fun part, right? 99.8% identical to our buddies, to chimps. The crazy thing, and the thing that's really cool, is that now that we have all these sequences, these genomes sequenced, we can actually look at what's different, right? And what, you know, so what's in that 0.2% that's so different, right? And a lot of it has to do with sensory perception, right? So with chimps, they've got insanely tuned hearing, and sense of smell and sense of taste. And we've actually lost a fair amount of that, right? But oh, what we got, brains, cooperation, language, all that kind of fun stuff, right? So what would you rather have? I don't know. Sometimes it's fun to smell things and hear things, right? Right, so those are, I mean, they're just like insanely tiny little differences, but yet in terms of sensory organs, they're actually very, very different. So that's kind of fun factoid, right? And what we've learned from the ENCODE project. So this is right, the Encyclopedia of something another. Encyclopedia. What does that stand for? Of DNA elements. Right, so the encyclopedia, the ENCODE project came out last year with all this sequencing, and we talked a little bit about that. Right, all of a sudden we found out that 80% of our genome, which we thought before was kind of junk DNA, didn't encode anything. Well, yeah, it's actually transcribed, and it makes RNAs. 9,000 of these small non-coding RNAs, including microRNAs, right? And 9,000 to 10,000 of these long non-coding RNAs, which we talked about a little bit, that are probably structurally significant and may have significance in terms of transcriptional regulation as well. This is another thing that actually to me is phenomenal, that there are almost 4 million sites within the genome where transcription factors bind, right? So at least has a capability of binding. So the transcription factor binding sites, right? 4 million transcription factor binding sites. That means about 40% of the DNA is actually accessible through promoter sequences, in essence, right? So that's a lot. That's a lot of the DNA that's essentially accessible. And there I have it. All I can say is, wow, that's pretty darn cool. So what's the rest of our genome, right? So 40% is accessible as transcriptional elements, RNA that's encoded, DNA that's encoded. What's the rest? Well, it's not junk. It's repetitive DNA, okay? And repetitive DNA, I gotta put my glasses on so I can read this. Sometimes it's structural, right? So we talked about telomeric regions and centromeric regions. Those are actually repetitive sequences, okay? They're, if you will, satellite regions. And we'll talk more about that in a second. But telomeres absolutely have a function of protecting the ends of our chromosomes. Centromeres, as we talked about, absolutely have a function. There are regions that are what we call moderate repeat regions, right? So ribosomal RNAs, tRNAs, housekeeping genes, they're all regions that are important and that are of varying lengths in terms of sequence, but that are oftentimes what we call tandemly arrayed. We need to draw a tandem array so that you know what that means. Okay? So when I talk about tandem arrays, this is what I mean. All in a row, we've got multiple copies of the same thing. Okay? So if this is 5 prime, and this is 3 prime of that piece of DNA, you've got copy number 1, copy number 2, 3, number 4, number 5, number 6. These can be arrayed genes, right? So ribosomal clusters, the 5S ribosomal cluster, we've got like 100 of these. These can be just repeat sequences, so satellite sequences like allo repeats. There can be lots and lots of these centromeric sequences. They're all arrayed like this. Telomeric repeats are all arrayed like this. So a tandem array just means that the same sequence is lined up one after the other. Okay, so in tandem. We're going to throw that word around a lot, so just so that you know what I'm talking about when I say it's tandemly arrayed. Okay, so why would you have copies like these?
Preservation of like valuable material.
Absolutely, right? So if I've got 6 copies of my tRNA, my F-met tRNA, my methionine tRNA, well, if one of them gets screwed up, who cares? I got 5 other ones that are going to be just fine, right? So genes that we need a ton of, particularly ribosomal RNAs and tRNAs, we have lots of copies, tandemly arrayed right next to each other, so that we know if one of them gets screwed up, we've got multiple copies to step in. Okay? Same kind of thing with housekeeping genes.
I don't know if you said this, but do these tandem arrays have stop codons in between, or does the whole thing get transferred?
Yes.
Oh, it does.
They have stop codons. Yep, absolutely. So this is an array. So with genes, right? With gene arrays, absolutely, each has a start and a stop sequence next to these. With satellite arrays, not the case, right? So something that's not going to be transcribed and then translated into a protein. It's just a repetitive sequence. That sequence might be arrayed in multiple times, but obviously since it's not being transcribed, it doesn't need to have start and stop. Okay, makes sense? Anyways? Okay, so what are satellites, VNTRs, ALU sequences, all of these things that are highly repetitive sequencing? So these guys can have a lot of different things that are associated with them. So what we call satellite regions, I can't really think of an analogy, so I'm not going to try. So satellites are areas of DNA that can vary in length. Okay, so we have what we call microsatellites, mini satellites, big satellites, right? It all depends on how big the sequence that is repeated is. Okay, so if I say I have a microsatellite, so for instance, the telomerase repeats, okay, the telomere repeats, TTAGGG, that's our repeat sequence. It is tandemly arrayed about 2,000 times at the end of our chromosomes, okay? So TTAGGG, 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 that's a microsatellite because it's only 6 nucleotides in length, and it's repeated over and over and over again. So microsatellites can be tandemly arrayed. Mini satellites, like in the centromeric region, can be tandemly arrayed, right? So the cool thing about centromeres, they're a mini satellite. They have almost the same sequence, but every single chromosome has a slight variation on that theme. And so the centromeres can actually be very easily recognized for each chromosome because the repeat sequence, the mini satellite repeat, is actually slightly different. So that's a really cool thing that you can utilize to actually mark the chromosomes, right? So remember when we looked at our sky chromosome spreads, well, you can do the same thing with just using the centromeric sequences. You can array those in a different color. And so each of the centromeres gets lit up by a different color. So we can actually see which chromosome we're looking at by what centromeric sequence we have. Okay. The things that we're going to talk about much more next week are called variable number of tandem repeat sequences. So these are also mini satellites. So VNTRs can range, they're roughly 20 to 50 nucleotides in length. And these are kind of cool sequences. They're all over our chromosomes, okay? So these VNTRs are all over the place. And they're regions that expand and contract, and each person is going to have a different variability of these, hence the name, variable number of tandem repeats, okay? So each person has an array of these VNTRs that is unique to them, except if you are an identical twin, then you have exactly the same VNTR patterning. So if you can get away with murder and then frame your twin, okay? Because, oh no, it's him, right? If you're really mad at them. But you shouldn't do that. So that's the cool part about VNTRs. They can actually be used for genetic analysis, for forensic analysis, because every single person has a unique set of VNTR lengths, okay? So VNTRs, and we'll see much more about this next week, are actually utilized in genomic screening to try and link genetic material to the presence or absence and the size of those VNTRs. So what we're going to do next week is we're going to look at how can we map a disease gene and follow the progression of a disease gene through a family by actually utilizing this VNTR sequence. So you inherit these, right? You inherit them from mom and dad, the length of the VNTR. And so we can track which chromosome we're looking at by looking at the length of the VNTR and seeing, well, how long was mom's? Oh, I got this chromosome from mom. And how long was dad's? Well, I got this chromosome from dad. And now we know if mom or dad has that disease, well, we know where it was localized because we can map it to the region where the VNTR is localized. It's a really cool technique. It's like crazy, insanely busy when you get it into really looking at multiple VNTRs. We'll take it down a notch and just kind of look at a couple. Okay, so, but, right, this is also allowing us to do forensic, what we call fingerprinting, right? So if you got a sample of DNA from Joey and a sample from John, they are going to look very different when we sample the VNTR sequences. And so we can see whose DNA came from where, and we can look to see, and this is used a lot in criminal analysis, whether or not this person that we're accusing actually did the crime. If their DNA was left at the scene, we'll probably be able to tell. Okay? So, cool things that can happen with VNTRs. Allorepeats are, again, interspersed all over the place on our chromosomes. They're actually a lot bigger. Allorepeats are about 300 base pairs long. So you could call that like a big satellite. And allorepeats can be a pain in the butt when you're trying to actually hybridize a probe to chromosomal DNA, because allorepeats, right, are everywhere. And so they bind up chromatin like crazy. They bind up chromosome probes like crazy. And so when you're doing analysis where you're trying to hybridize a very specific site, a very specific piece of DNA to a large amount of chromatin, you actually have to block the allorepeats so that your probe doesn't stick non-specifically onto this allo sequence. It's a pain in the butt, and it also is problematic, right? Because think about this. Allorepeats are interspersed all over the place on our chromatin. And they have pretty similar sequences. They're not identical, all the allos, but they have very similar sequences that are interspersed all over the place. So, right, when we're trying to do crossovers, meiotic crossovers, or in a situation where we're not tightly controlling crossing over events, we could potentially have non-accurate crossover between two chromosomes at these allo sequences, right? So remember our sky, our paint, where in the tumor cell we had all these crazy crossovers occurring. Those are most likely occurring during at allorepeats, where we've got an allorepeat on chromosome 3 and an allorepeat over here on chromosome 7. Oh, they look kind of alike. Let's cross over here, and now all of a sudden we've got a chromosome 3 attached to a chromosome 7 p-arm. Okay, so allorepeats can be very dangerous if we don't have proper surveillance of the recombination machine. Luckily, we have that in general. And then lines and signs are the last guys I want to just hit on, because as a virologist, these are insanely cool to me. Right? So lines and signs are, again, these big repetitive sequences, motifs that we have interspersed throughout our chromatin, okay, throughout our genome, and they are the remnants of interactions with retroviruses many, many, many millions of years ago. Okay? The cool part is, again, to me, virologists, cool part, we actually have some of these in our system that work. And that work, and by working, I mean they actually produce virus. Right? So they're retroviruses. They're encoded in our DNA, and they have all of the components to actually make virus. And we shed endogenous retroviruses. They're not hurting us. They're not problematic. They're not jumping around all over the place. And we're shedding them, and the cool part is that they are actually evolutionarily helpful to us. Okay? And so I want to just gibber off a couple of examples of human endogenous retroviruses that are helpful to us. And I think I put this in there, but if I didn't, I'll in my updated version, I'll throw this in so that you have this information. So there are 3 curves that I have examples of. So they actually serve in an evolutionary sense as intracellular restriction factors, and also potentially as receptors on the surface of cells. So a really good example of the latter is that there's one of our endogenous retroviruses where its glycoproteins are actually presented on the surface of a cell called the syncytia trophoblast. So syncytia trophoblasts are part of the placenta, and they're part of the placenta that invades into the mom's uterine wall and helps to attach it, right? And the syncytia trophoblasts are these cells that are crazy mobile, and they're just running all around all over the place, and they're making these ginormous syncytia, right? So they're individual cells, and then they fuse to form enormous syncytia that then invade into the uterine lining. Okay, and so syncytia trophoblasts have glycoproteins on their surface that help them to bind and navigate into the uterine wall. And those glycoproteins are derived from these endogenous retroviruses. Cool? Yeah. Second, there's tissue-specific expression of this protein, or this enzyme called salivary gland amylase, right? So it's only expressed in salivary glands. And that's because the endogenous retrovirus insertion points controls the expression so that it's only expressed in salivary glands and not expressed in other glands. How cool is that, right? So it's actually evolutionarily evolving with us over time to have tissue-specific expression, syncytial trophoblasts, salivary glands, and also pluripotency. So there's an endogenous retrovirus in ES cells, in embryonic stem cells, that actually keeps their pluripotency, and the retrovirus is there and helps to continue to have it be pluripotent. Nick, did you have a question?
Yeah, I just was trying to wrap my head around it. So we have this strand of DNA
Good luck with that.
that codes for this retrovirus.
Mhm.
Is there a strand of DNA next to that gene that would code for the RNA that they would then have to use to make DNA? I mean, maybe I'm getting...
So the retroviral structure, so it's a double-stranded DNA when it's inserted into the cellular DNA, okay? And there are control elements on either side, right? So let's look at this for a second, and we'll talk more about this tomorrow. So retroviruses are crazy birds, okay? And they're incredibly compact, but they encode an enormous amount of genetic material that is overlapping, and they're polyproteins. Okay, so the gag, pollen, envelope proteins are all encoded within the boundaries of the retrovirus. So there are regions on the ends of the retrovirus called long terminal repeats, and those are the control regions for the virus. Okay, so all of the encoding genetic material is inside those long terminal repeats. Okay, and the long terminal repeats are where it inserts into the cellular genome. Okay, so depending upon whether all of this sequence is still present or just parts of it are present, gives you whether this thing is an endogenous retrovirus that can actually be expressed, or a line, or a sign. So depending on whether these LTRs are still present, or whether you've got still the presence of some of the viral protein sequence, is going to give you whether you're a retrotransposon, or a line, or a sign, or an actual whole endogenous virus. Okay, so to I think answer your question, Nicholas, the inserted material is DNA, double-stranded DNA. And so there are control elements for the virus if you've got an entire retrovirus inserted, or if you've got a line or a sign, those elements, the control elements have actually been deleted over time, over evolution, coevolution. But these purple sequences here are repeat sequences that, how to explain this, occur as you insert the DNA, the double-stranded DNA, into the site where the insertion is occurring. So you actually get a small replication event on the ends of the cellular DNA where the retrovirus inserts. So that's actually where you can tell where that retrovirus has inserted, is because you get a little tiny duplication of the cellular sequence on either side. So it may be that the whole thing is there. It may be that part of it's there and the LTRs are still there. It may be that most of the sequence is gone, but you still have some of the genetic material intact, and you have those repetitive sequences in the cellular DNA that are still there. So it depends completely upon whoever you're looking at. So here, right, with the endogenous retroviruses, just a little retroviral info. So the GAG are the some of the structural proteins. The polymerase is where the reverse transcriptase and the insertion proteins are. And the envelope, as the name implies, are the envelope proteins, the glycoproteins. Okay? So if you don't have envelope, you can make all the retrovirus you want and you can't go anywhere, right? Because you don't have any glycoproteins to actually leave the cell. The lines and the signs at this point have lost the genetic material that encodes even the polymerase function, but they still have the LTR sequence some degree. Some of them do. What's cool about lines is that they actually are still mobile. So this is a kind of crazy phenomenon, and can be a little scary when you think about it. But these elements are what we call mobile elements. So they can actually jump around in our DNA. They're called transposons, if you will. And for the most part, the lines and the signs are clamped down. So this sequence is highly methylated next to where the jumping would occur. So methylation is going to actually silence the DNA, and we'll talk more about that later. But so the regions where the jumping would occur, where we would excise ourselves, are silenced in most of the line and sign material. But there is a set of lines called L1, which actually still can jump around. And there are about 100 copies that can move around still inside of our DNA. And the cool part about this, and this is a guy that is down at Scripps, at the Salm, down at UC San Diego, where I did my graduate work, and Rusty Gage has studied these lines in neural development. And so the really super cool part about this is that these lines are really active in neurons. And so it's thought that the lines and the jumping around that the lines can do is actually providing genetic diversity during development of neurons. And that particularly during neural development, this is allowing the neural landscape to morph and to kind of adapt to different cues that it's receiving. And that these line elements are thought to actually be helping to provide genetic diversity and to provide diversity of response inside the neural cues. And when you knock them out and you don't allow them to jump, then you get a much less diverse population of neurons during development. So that's kind of a cool process. That, again, right, who in the world would think that that's what's going on? But it is, in fact, cool. And, right, if we didn't like this DNA, we would have gotten rid of it a long time ago, right? If it wasn't beneficial, if it wasn't something that was going to help us and that we've co-evolved with, we wouldn't co-evolve, right? We would have dumped it out a long time ago. And that's the thing that you got to kind of keep in your heads, right? This isn't junk DNA. We don't have junk DNA. Everything's got a purpose. We're just not sure what it is yet. and that's the thing that we need to just keep in our heads. Now, all right, yay! This is the end of topic 1, right? We had our goals. We've thought about it. And since we only have 10 minutes, and that's silly to start something new in 10 minutes, we're going to have some fun. And we're going to do something fun. Yay! So I want you to break up into little groups. Groups of, how many are we? 30, 35, 30, 30, however. Okay, I want you to group into groups of 5. 5 or 6, it doesn't matter. that you thought were the coolest, or the most important, or the thing that you were so most surprised about in this lesson? The top 3 things. And then we're going to write them on the board and we're going to vote. Because voting is good. Yeah. Topic 1. Things in topic 1.
It was 5, yeah. It's an easy group.
Hey, you got 5 minutes.
I thought endogenous retroviruses was really neat.
Yeah, at least the evolution of it, how we've worked it into our cellular function. That's pretty amazing.
Yeah.
It's akin to how we worked, mitochondria have very similar genetic makeup to